We're going to talk about this concept of secondary endosymbiosis. To make it very simple and then go into the complexity of it is one bacterium engulfed something that allowed it to photosynthesize. And then that bacterium was ingested into another the second time, and then you have this diversity where you have something that is may be able to photosynthesize or may be able to do something that belongs to the property of the original eukaryote that was taken in. And so we'll go into it a little bit more specific. So what's happening is you have more diversity in the final like the final dinoflagellate because of the fact that it was created by one plant engulfing or one species engulfing another. So you have the original cyanobacterium, which is a bacterium that was able to photosynthesize. That engulfed over the course of evolution, engulfed something that lost its membrane, and then that membrane actually then was incorporated into the cyanobacterium. That losing of the membrane is where we get red and green algae. Red and green algae is our eukaryotic species that can photosynthesize. From the red and green algae, you have this secondary endosymbiosis. And then with regard to the secondary embiosis, you have the ability to create these plants called chlorarachnophytes. You see that word at the bottom of the page, chlorarachnophytes. And they evolved when heterotroph eukaryotic cells engulfed the green algae. They know this because and so you have a portion of it that has chlorophyll in it, as well as the next one up that has a plastid that we know came from another organism other than this, but it's this photosynthetic property. But yet you also have membrane-bound organelles, and you have other functions with regard to it. So endosymbiosis both benefit, and what's happening here is you had one symbiosis taking place, endosymbiosis taking place, and then a secondary one, and ended up with a species that's much more complex than any of the beginning. Here we are with the supergroups again, just to remind you that that's what this chapter is about. There's a few terms like endosymbiosis that are to be understood, but most of what this chapter is about the 4 supergroups. Here you have the 5 listed because the last 2, the amoebas and then the fungi, are broken apart, but in your book those are put together into just one group. But the idea is to identify the 4 supergroups and some of the key characteristics of each group. Moving on with that idea, and I'll try to bring that image back each time we move through a supergroup so that you remember that that's what you're learning this stuff for, or being exposed to it. Excavates are protists that have a modified mitochondria, but they also have this unique flagella. They have a cytoskeleton, but most protists are characterized by their cytoskeleton. It's something that I forgot to say in the very beginning when I introduced you to protists. The advantage of a cytoskeleton, which eukaryotic cells don't have, is you're able to have numerous different, diversity in structure, diversity in size and shape, but mostly shape. And also there's some organization inside the cell that you don't find in prokaryotes because of the cytoskeleton. These have a feeding groove. That's one of their unique features, this feeding groove, which will eventually becomes a mouth. They have the beginning of an eye spot, so what they have is some cells that have a receptor type cell in there, and that also gives them an orientation in terms of up, down, or what we think of as face and non-face. There are chloroplasts, and then one other thing that you find in this particular supergroup, as well as some other supergroups, is this contractile vacuole. And what can happen, this contractile vacuole can do is not only allow this this organism to live in a fluid environment, even though it's going to take in fluid, because it can actually expel the fluid out via its contractile vacuole, but the contractile vacuole can also help it move. And so feeding groove does have a flagellum, a little bit of an eye spot, but the big thing probably is that feeding groove. They do have a mitochondria, but it's not a well-developed one. We call it a mitostomes, so not quite, 2 haploid nuclei, and a flagella. Other excavatas, 2 other groups that live in anaerobic environment, so not in an aerobic environment. You have diplomonoids, and I think that's probably diplomonads. They have, again, the modified mitochondria. They can take energy in via glucose, so glycolysis, again 2 equal nuclei, and multiple flagella. So this one, multiple flagella so far. So flagella and the modified nuclei, modified mitochondria, tend to be 2 things to hold on to with this particular supergroup. Again, we're in excavata. Then the next group also reduce mitochondria and also generate energy anaerobically without oxygen. And one of them in this group is the pathogen that causes yeast infection in females, in terms of problems, I guess, or ones that you might be familiar with. Again, you see the word excavata over there, so you're putting multiple flagella and reduced mitochondria in with that particular group. Third examples of excavata, euglenozoa. Sorry about that, it's a mouthful. This is a clade, and that's a group or a subgroup. You can think of a glade as a subgroup that is predatory. And also photosynthetic, also autotrophs, mixotrophs, and parasites. So you have a little bit of everything there. And one of the distinguishing features is a spiral or a crystalline rod inside their flagella. They don't know really what it's for, but it is found in their flagella, so a unique flagella with this group. But again, flagella is the key word. This group can photosynthesize, though. The next group, Clintoplastids. This group has a large mitochondria or organized massive DNA, and that's called a clintoplast. They are free-living consumers of prokaryotes in freshwater, and this is the eukaryote that causes sleeping sickness, Chagas disease. and a couple of other illnesses, but sleeping sickness in humans. And then if you look at the picture on the right, you see these clinoplastids in blood cells. Those are red blood cells. And then you see the kinetoplast, that unique mass of DNA, also a nucleus in the cell itself. Again, I apologize for the pronunciation, but it's all a tongue twister here. You aren't expected to learn how to say them unless you're just a master of that that particular skill, but you do want to kind of recognize the word if you see it on a multiple choice test. And the euglenids, very similar to what we saw before. They have two flagella that emerge from a side pocket in one end of the cell, and some species are, can be mixotrophs. You also have your contractile vacuole, as well as a light detector. These are basically cells that, again, as I said, that have photosynthetic ability to capture light. Supergroup again, because we're going to move on to the next group, and that's the alveolates. They again, and another thing that happens here is sometimes you have a slightly different way that the group is spoken about, but your book calls these the alveolates, the SAR group, SAR group. And here we have this particular, the SAR, taken from the beginning letter of Starmenophia, the alveolates, and Rhizara. Those three groups make up the SAR group. That's the next supergroup. So we've only moved on to the second supergroup. And this is pretty kind of an interesting group, particularly some of those images you see of very, very colorful, very pretty eukaryotes. This tends to be the group they come from. Characteristics: secondary endosymbiosis of red algae here, and then also similar DNA sequences among the entire group, and that's why they're grouped together. So it's the SAR clade. First in the SAR clade are the staminophiles, and that includes diatoms. Those are those glass-walled silica, very pretty. You see those in ocean pictures all the time. Brown algae and some water molds, but brown algae and golden algae in this particular group. Here you have a piece of seaweed and the parts of the seaweed. Very interesting in terms of adapted. Seaweed is adapted to grow in the ocean. And what you have are the holdfast is what keeps this seaweed attached to either the it grows in the margins in the ocean, but attached at I guess a bottom portion. I don't want to say the bottom of the ocean because that's not necessarily where they're attached. There's a stalk type where you have a root-like holdfast, and that anchors, and that basically is what we were just talking, the root-like holdfast. It's not a root, but it's root-like. A stem-like structure called a stipe, and then leaf-like structure called blades. So they aren't quite that, but they are similar to what we recognize as plants, and certainly kind of look like plants. We'll pick this up on the next one.